When discussing genetic circuits, we saw many designs involving transcription factors and other proteins. It is also possible to construct genetic circuits using microRNAs in eukaryotes. This study by Renato et al. Demonstrates logic gates that operate in mammalian cells based on siRNAs. Logic gates that operate in cells are a popular target for synthetic biology. Mathematically, a large set of orthogonal inverters would be sufficient to form any type of Boolean logic. Essentially, this would be the biological equivalent of having many transistors operating in the cell. The primary reason for doing this would be to construct signal integrators that merge multiple sensory inputs and respond with appropriate actuation responses. Mechanistically, any chemical strategy in which expression of an RNA or protein controls the expression of another of its own kind could be used in scalable logic gates as long as the large orthogonal set of these biomolecules are accessible. Others have pursued strategies involving recombinases or transcription factors amongst other routes. In this paper, the authors work towards the goal of large integrated genetic circuits using siRNA-based designs in mammalian cells. First, let me explain their diagrams. The yellow shape is a yellow fluorescent protein under a constitutive promoter. There is a recognition sequence for an siRNA on the 3' end of the YFP transcript. Cleavage of this sequence will chop off the poly A tail, inactivating the mRNA. The dotted circle is some sort of input that gives rise to production of the brown box. This brown box is the guide RNA RISC complex in the cell. Thus, the presence of the A input results in formation of the active complex and knockdown of the YFP gene. In their experiments, the inputs like A are transfected siRNAs, but they could also be encoded genetically as microRNAs. In this circuit, the siRNA controls a LAC-I gene instead of YFP. LAC-I then goes on to transcriptionally control an RFP gene. This is ultimately a NOT gate. Here, two siRNAs control the LAC-I gene, resulting in an OR gate. They also can express more complex logic. Here there are two copies of the gene encoding LAC-I. The first copy is an OR gate of siRNAs A, C, and E. The second copy is an OR gate of an siRNA inhibited by A and B. The result is the complicated Boolean statement A or C or E and not A or B. One nice thing about engineering with siRNA is that the genes are quite small. There are a large number of interactions present in the system, but sequence-wise, an interaction only involves a 21 base pair sequence in the mRNA.